Ladies and gentlemen, a very good morning to you. Let me first welcome Dr. Diana, the Science Counselor in the Embassy of Hungary in India. She is a great friend of India and of Amity. We are thankful to her. For so kindly bringing two distinguished agricultural scientists from Hungary to Amity. I welcome Professor Thomas Montessori and Dr. Melinda Tarr to Amity and look forward to their lectures and deliberations with Amity scientists and researchers. May I now request Dr. Doyle Sanyal to kindly introduce the distinguished scientists.
Thank you, Captain Shukla. We have two speakers today from Szeged University, Dr. Thomas Montessori and Melinda Tarr. Thomas Montessori, he has done his master's in agriculture and now a college professor and is heading the Institute of Plant Science and Environmental Protection at the Faculty of Agriculture of the University of Szeged. Basically, he graduated in 1991 at the Godolo University of Agricultural Sciences, and after 2 years of working in the Central Civil Research Institute, he started his work at his current place, Szeged University. So between 1991 and 2001, he was working for 2 years in the Institute of Plant Biochemistry in the research group of Bettina House, aiming to modify endogenous jasmonate level in barley through introducing AOS1 and cDNA in sense and antisense position via particle bombardment. Now, his PhD thesis was on microspore culture and genetic transformation studies in barley and triticale in 2003. So he made his habilitation in 2022. So the topics of his lecture and his scientific presentation focus on sweet potato. His main course taught are plant production, breeding, agriculture biotechnology, fish farming, both in Hungarian and English language. In the course of his career, his research work included plant biotechnology, especially in vitro tissue culture, plant molecular biology and gene technology, plant production, small grain cereals, dry bean, potato, sweet potato, and fish farming. Since 2015, he is focusing on sweet potato, continuously having PhD students making research on the topic from different species, cultivation methods, nutrition, conditioning of stress resistance, in vitro micropropagation, etc. In the last years, he returned to his research field from university, that is fish farming. He primarily focuses now on the effects of feed sources on the development of fish fry and fingerlings. I'll now introduce Dr. Melinda Tarr from University of Szeged. She is a senior agricultural researcher and plant scientist with more than 20 years of experience in crop science, plant breeding, pathology, sustainable agriculture technologies. She currently works as a senior research fellow at the Faculty of Agriculture, where her research focuses on legume production technologies, nutrient management used by biofertilizers, effects of different intensities of tillage on soil-plant microbe interaction, and the effects of beneficial microorganisms on the plant crops. Previously, she held leading research and management positions at Hungarian University of Agriculture and Life Sciences and National Agricultural Research and Innovation Center. Dr. Tar has led or contributed to numerous national and international R&D projects related to sustainable crop production, soybean cultivation, biomass utilization, and climate adaptation in agriculture. In addition to her research activities, she is actively involved in higher education through teaching, supervising PhD students, curriculum development, and delivering lectures in both Hungarian and English. She has authored 128 publications and serves on several professional and scientific committees, including Hungarian Plant Breeders Association. We welcome both of you, Professor Tamás and Dr. Melinda.
Thank you, Dr. Sanyal, for the introductions. Before we begin the formal deliberations for the day, may I request Dr. Nutan Kaushikji to say a few words and invite the distinguished professors to deliver their specialist lectures.
Thank you, sir. Very good morning to all of you. On behalf of our Honorable Founder President, Dr. Ashok A. Chauhan, our Chancellor, Dr. Atul Chauhan, sir, our Vice Chancellor, Ma'am, Dr. Balbita Shukla, ma'am, on behalf of the entire Amity universe and on my personal behalf, I welcome you all to this very important event, which is the visit of the delegation from University of Szeged from Hungary. We were looking forward to your visit. The day we had a meeting with Diana ji last time when she has interacted with all of us, and then she informed that two of the scientists will be visiting soon in the field of agriculture. Can we arrange the visit? And we were so excited to listen to that. That yes, the outcome is just in the offing. So, so we that happily accepted, and very quickly Diana ji to both of them, and then they also agreed to give the guest lecture in such a short notice. So we are so thankful to both of you for agreeing to give the the guest lecture despite being just told 2 days before. So that's so kind of you, and we are looking forward to the interaction. I would just like to say that the topics which you have chosen are very important topics, and Amity is very much aligned with the topic, the presentation which you are going to give today. And we have, later on, I will be giving a brief presentation about the activities being done in the field of agriculture and the right sector, so that we can have meaningful interaction later on to develop the collaborative activities. The one which, Professor Thomas Monsari is has diverse, I think, experience. I was listening to his brief CV when Dr. Doyle was presenting it, and I was thinking that his experience is just like the experience of mine, diversifying from plant protection to breeding to bioprospection to medicinal plants and to fish also. And I have also worked in almost all the sectors. So I was very much impressed the diverse areas which you have covered. And probably during the presentation, we find some of those glimpses which I would be able to give. Like I started with the what you call the breeding of the double low varieties. So I was helping the breeders in monitoring their seeds for the two important characters, that is oleic acid, that is erucic acid in the oil, and the glucosinates in the cake. So how to screen so many samples? There were thousands and thousands samples to be screened in 2 months. So we had to develop very quick and sharp method like the half seed method to fix the early characters, and also the single gene analysis and so on. So I'm very happy that you also have a similar experience. Similarly, Professor Belinda Thar, she has worked on legumes, biofertilizer, and soil health, which is also very important for climate resilient agriculture. And we are very much aligned with. We have many scientists working in this area. So probably the interaction which we have today will have a very fruitful outcome. I think Director Outcome is sitting here. So we will make sure that whatever we discuss here and we bring the fruits are seen in near future. Thank you so much. So we welcome you all here. And I request everyone to be open for the discussion later on after the guest lecture, and ask as many questions, and look forward to the opportunity which you can collaborate with them. Thank you so much.
May I now request Professor Thomas to kindly give his guest lecture. So, dear colleagues, first of all, I would like to thank you and especially the organizers to give us the opportunity to introduce us and to meet you. And we hope that after numbers of collaboration with other colleagues from our university, we can start a collaboration in the field of agriculture too. So as you heard from the brief introduction, in the last 10 years, I've been working on sweet potato especially, except for fish. And that's why I've chosen this topic for my short presentation. I try to keep time because we shared 1 hour devoted for us with Dr. Melinda, and I would like to leave her enough time for her presentation. So sweet potato is very familiar to you because it's by origin a tropical, subtropical crop. Just to make some refreshing of your knowledge about sweet potato. So it's a very important food crop in the world. It's got a very impressive nutritional value, high energy content, and especially the orange-pledged genotypes are a very remarkable source of beta carotene. So I managed to visit the potato center in Lima, and the director general told me about their program to distribute the orange-pledged sweet potato in the sub-Saharan region to exchange the currently existing and well-known white-fledged ones to introduction of beta-carotene deeper. It improves blood sugar control very well, but be careful because although its glycemic index is moderate, but after heat treatment it increases. It's a very adaptable crop, even in Hungary. So under
not the natural conditions for considering the origin, but it works very well. So it's not that interesting. Its foliage is also very important in some African countries. They cultivate sweet potato, especially for the foliage and the leaves, and less for the storage roots. Its quality is similar to that of spinach. It can be used as animal feed, and in industrial utilization, there are also perspectives, but in the developed countries, it is still the situation that it is more economic to use it as a human food, and less as to process it for ethanol, for example. Here you can see the world continents. Unfortunately, FAO doesn't register the European cultivation since 2018. In almost all the European countries is sweet potato grown, not only in these four that were registered at them. In the world, China is the first, and it takes from the area 30%, and from the production 55%. But in the yield potential, Guyana is the first, but I don't think that it comes from one crop. This result would be impossible, so it must come from 2 crops per year. So, but it wasn't communicated in the FAO register. In Hungary, sweet potato is known, not well known, but at least known for more than a century. There were experiments on sweet potato before the First World War, and then it was interrupted. Then in other sites, it started during the communist era, but the recent sweet potato area started in the middle of the 80s with one whitish fleshed cultivar and one orange cultivar that was developed by our fellow breeders at a family farm. Its taxonomy, it's well known, so it is not a close relative of potato. It belongs to Convolvulaceae family. The gene center is somewhere in America, Central America, and South America, but it was distributed early. And very important information is that is never communicated by Greenpeace activists and other anti-GMO guys, that sweet potato is a natural GMO that is not just including but even expressing genes from Agrobacterium tumefaciens. So it is important information. Regarding it likes the warm temperature, precipitation, especially in the early times, light soil, rotation, soil preparation can be done flat or making ridges. Nutrient supply, to mention, it is potassium demanding crop. Planting can be done only if the temperature is high enough. And so here you can see some pictures from Hungary. And cuttings are used, especially primary cuttings, but secondaries can also, or cuttings in soil block. And in tropical, subtropical countries, mainly cuttings from cut vines, vine cuts can be used. Manual planting is possible, and also mechanical machine planting, like in Portugal here and in the center in Hungary. And irrigation is needed, but sometimes they provide a short abandoning of irrigation after 50, 60 days to promote the development of storage routes. And drip irrigation and sprinkling are also available. Regarding the plant protection, weed control is necessary just in the first period. After that, it suppresses weeds. It can be dangerous, especially if they come through the planting materials. So virus-free planting materials is a must. Soil must be disinfected against soil-borne pests, and harvesting can be done either at biological or at technical ripening, and can be done manually or by potato harvesting machines or seedling undercutters, lifted and collected manually. So much about briefly about the well-known things. The current status of sweet potato in production in Hungary is that there is a continuously increasing demand for sweet potato, and as it is normal, it stimulates the producer's interest. But even at such producers that have a long time experience in producing sweet potato, they've got problems with the yield stability. And so there's a contradiction, and this must be solved. And that's why more than 50 years ago, the colleagues from the sweet potato family farm came to us to help them. At that time, they had much more experience in sweet potato compared to us, but we tried to develop. Here is a list what should be solved. So site and cultivar specific technology must be very important, is to provide safe planting material for the producers based on in vitro micropropagation. And so by these the yield and the crop stability will improve, and that can help to increase the production area and total production, and we can reach that Hungary doesn't need import from sweet potato. I don't think that it will happen in the next decades. So we currently we harvest the sweet potato by not later than October, but in May there is already no Hungarian sweet potato available on the market. So we import it from foreign countries, even from US, but from Egypt, Israel, and such countries. But when I visited Israel, it was interesting that I asked them to show me sweet potato and the technology, and they told me in advance that they won't communicate to me all the secrets, because we are competitors in the market, because they export sweet potato to Hungary. It was a bit strange, but anyway, okay, they were kind guys. So we finished a project recently to improve the sweet potato technology. The goals were those that I mentioned as the main to-dos and main improvements in necessary improvements in Hungarian sweet potato production, and now some results from our experiments. So we were interested in how much the planting time can influence the sweet potato yield together with the harvest time. So because in Hungary we can start planting of sweet potato in the late May period, and it seems to be economic to do it till the end of June. In July it is not good, but our experiments showed that the early planting together with the early harvesting, so keep the growth period the same, approximately 120 days. So the result was the best if it was planted early and harvested early. And compared to the first result, maybe these data are a bit exaggerating, but the difference could be quite a lot on hectare level. We were interested whether it is worth to make ridges or not. In the recent in Hungary, first everyone planted sweet potato flat, so without ridges. But in the last some years, almost everyone in ridges covered by foil, and so on sandy soil, however, our result showed that flat planting can be economic. Not much better, but it works. So on sandy soil, you need not invest much time and effort to prepare ridges. Anyway, it is not easy to prepare them on sandy soil, but on hard soil, it is better to form ridges. We were interested also in the optimization of the plant density, because the colonized setup was 100-meter row distance and 30 centimeters plant-to-plant distance. Regardless of the cultivar, we were working with one of the Hungarian registered cultivars, the AUUP 12, and we showed that 80 centimeters row distance are enough, together with 20 centimeters plant-to-plant distance. And even making an economic calculation, because such a denser population needs more cuttings that cost quite a lot, because sweet potato planting material is not cheap. But even considering that, the higher yield will equilibrate the yield. So it is economic to make this dense population, at least at this cultivar. We tended to work with this registered cultivar because then we could be economically and from the low side correct. We tried the greenhouse growing during the whole growth period, but it gave very poor results. So the best result was 350 some grams per plant, and tell the expectations under Hungarian conditions, we say that we are satisfied with the yield if 1 kg is the yield per plant. So it was far below our expectations. Actually, we expected such a result. So our previous not correctly set up experiments showed that it cannot be economic. So maybe in the early period it's okay, but for the full season not. And the rest was the purple. And we experimenting with various nutrient sources, and in this large experiment, we inserted a bacterial inoculant among different versions of chemical fertilizers. And okay, so I don't know the details. And the soil was sandy soil with relatively good nutrient supply. But anyway, one of the chemical fertilizers gave the best result, and the second one the second best. But the result achieved with the bacterial fertilizer alone, without supplementing with anything else, was quite important. When we classified the tuberous, that gave also very good result, and we showed that the application of this bacterial fertilizer made the plant's organic matter building up capacity better. So it could achieve very good result parallel with building the lower foliar mass. So the difference between the best result and the bacterial fertilizer result was merely half tons per hectare, and that suggests that to work with this bacterial fertilizer alone can be economic, especially if you are in the organic cultivation business. Yeah. Then we tried experiment of bacterial microbe-based fertilizer that was developed by the microbiologists of our university. And here you can see the components. It was not merely bacterium-based, but trichodermas were also involved. And first we tested the best treatment. It was the best when we soaked the slips into this complex, and the best result was given when an extra soil inoculation was applied, and the average storage root weight was also the best with this treatment. But in the next experiment, we compared the efficiency of this mixture with the normal technology, farm technology, and it was very interesting that even the worst result achieved with this microbe complex gave better result than the farm technology based on chemical fertilizers. Yeah, so we concluded that these microbe-based soil inoculants can efficiently replace traditional fertilizers, and different compositions can be efficiently used. You can see what were the microbes involved in our experiments, and in literature what else can be found. One of my PhD students performed a very nice experiment. Here you can see the title of the paper that was issued. The characterization of the endophytic and the rhizospheric Bacillus licheniformis strains is isolated from sweet potato with plant growth promoting and yield enhancing potential. There are numbers of experiments communicated. Here I copied just one table that shows the first step of choosing this 27715 strain, and you can see that how nice result was given by this strain in field compared to the control without any treatment. Yes, we made 2 years experiment whether it is necessary or worth to lift the sweet potato vines during cultivation to avoid the building of small tubers or at least roots at the nodes contacting the soil, but no, nothing exact result. We gained, so in one of the years, the lifting, other year, the non-lifting gave better results. So it's up to you. We made still make in vitro experiments also, and we tried the MS medium without any hormone supplement that works very fine in practically all the cultivars we examined, but in some of them the supplementation with BAP gives better results. You can see that you have to test which is the best composition of culture media for your cultivar, so it must be done always. Microsilo experiments were also performed at our faculty. Actually, the results are still not convincing. In the first year, some of the tanks went wrong, but the non-rotting ones gave promising results, but it is still not sure to be communicated the results. And what are our conclusions? Not necessarily based on our experimental results. So there is still a lot to do for making Hungarian sweet potato supply independent from imported tubers and planting material. Here, an acknowledgement to our colleagues involved in this work. I've got now one PhD student from Ghana working on sweet potato. I've got 2 already defended his and her PhD. And thank you for your attention, and give the floor to Dr. Melinda. Thank you.
Thank you so much, Professor Thomas. I would just like to share something interesting. In India, the potato is called aloo and sweet potato is called kachalu. So unlike in Europe where it is a staple food, in India, sweet potato is normally used as a snack, especially in winter season when it grows. And I remember as young boys and all, we used to barbecue the sweet potatoes and peel off the skin and then put a little bit of rock salt on it and then some lime and eat it as a snack. And it is very popular with the ladies and children in India. So I suppose you can guide our agriculture scientists. This crowd is of agriculture scientists and students, so that we can also export sweet potatoes to Europe when it is required. Thank you so much. Now, may I request Dr. Melinda to kindly address the gathering and deliver her lecture.
Thank you very much.
Good morning, everyone. First, I would like to thank for the invitation and for the introduction. And today I will present you the effects of biofertilizers, especially arbuscular mycorrhiza fungi on plant health and production and soil health, including results of my own experiments. So nowadays, the soil cultivation and with it crop production is facing a paradigm shift for many reasons. The soil-plant relationships is usually understood as one-sided, like the plant taking water and nutrients from the soil, but this is far from the reality. This relationship is much more complex and especially for the plant and also especially for the soil and the soil living organisms. This is a cooperation and it's very important. There is a very big question. Is it dirt? The soil, is it dirt or not? Why is it so important? And why is it important to know about the soil microorganisms and also for the plant soil microorganism connection together? I think everybody thinking about that, how many living organisms are under our feet. In worldwide known now that 1 teaspoon of soil contains more living organisms than there are people in the world. So it's a huge amount. And if I'm thinking about it, we can ask help for them, for plant production and for our economy. There are several different taxa of bacterial cells, and in 1 teaspoon of soils there are about 200 meters of fungal hyphae, and beside of the macro and microorganisms, the other important components are the roots of plants. The plants and especially the plant roots are the key factors of this soil food web, because there are plants which have deep or shallow roots, or main or lateral roots, or thin or thick roots. So on the picture you can see these several type of roots, and they also can help for us to make connect between the microorganisms and also the plants. Around the roots, we know that there are a teeny space of special underground neighborhood where the plants and microbes lives. The name of this that is rhizosphere. The narrow region of soil are on the roots, and these microorganisms live, talk, and trade on this rhizosphere, and make connection between the plant roots and the plant and the soil. Up to now, we know that more than 140,000 fungal species have been written, but it should be a small part of the real number are known. And now we know that there are some pathogen groups which can affect the plants. There are decomposers which can help us to give micronutrients and macronutrients of the plants. There are several types which are plant growth promoting, which are promoting the plant growing, the micronutrients growing uptake, and there are the arbuscular mycorrhiza fungi with special symbiosis together with the plants. What does it mean, mycorrhiza? Mycorrhiza literally mean fungus, root. Arbuscular mycorrhiza fungi, the symbiosis is
on both sides, the plant side and also the fungi sides. It's very important, and the connection is also very specific because the plant give for the fungi sugars, and the fungi give for the plants several mineral nutrients and also water from the soil. We know that this symbiotic association, it's over 80% of plant species engaged, but in some forms of mycorrhiza symbiosis. It also promotes the biodiversity and ecosystem resilience of the plants and also in the soil. Over 400 million years ago, the plants started to move from the water to the dry land, and this dry land was very hard for the plants because they had no root yet, and they cannot uptake the nutrients from the new ecosystem, new soil, and also the soil was very poor in nutrients, so the water and nutrient uptake was hard to the plants. But how did the plants can survive this period? They didn't do alone. They do it together with these mycorrhizal fungi, and these fungi live in the soil and gave micronutrients and waters for these new dryland plants. There are thousands of species of mycorrhizal fungi. You can see on this picture that there are specifics for ectendomycorrhizas for trees, or ectomycorrhiza also for forest trees, orchid mycorrhizas for orchids, ericoid mycorrhizas for ericaceae, and the arbuscular mycorrhiza for several plants, several agricultural plants. As I said before, the most important type of mycorrhiza in agriculture, the arbuscular mycorrhiza. This is a very special group of fungi from the Glomeromycotina group. These are obligate biotrophs, so they only can live together with the plants, and less species are partner specific. We know now, up to now, 200, 300 species on many places in the world. And the fungi, if they are present on the root surfaces, has individual traits and may penetrate directly into the root hairs, as you can see on the picture. The plant partner, almost every plant group can connect to the mycorrhiza, but there are several from the Brassicaceae and Amarantaceae groups, which classic non-mycorrhizal plants are in these groups. The mycorrhiza can help for the plants in the phosphorus uptake, and on this picture you can see the main structure, so the biological overview of arbuscular mycorrhiza. You can see the vesicles, which can storage the nutrients, the micronutrients, the arbuscles, which is tree-like, and it is in root cells. It can help for the nutrient exchange between the plant and the fungi. Also, we can see the intracellular hyphae and the spores in the soil, and it's very easy to see under the microscope, because we are using very simple but effective method with blue penning and vinegar to dye these mycorrhiza and check under the microscope. On this picture you can see soybean roots, mycorrhizal soybean roots, with different arbuscules of mycorrhiza. The role of this arbuscular mycorrhiza summarized on this picture. You can see that the defense against the pathogen and the mycorrhiza increases the resistance to the drought of the plant. So the activating plant immune response is very important trait of this fungi. Also, as I say for you, it can improve the nutrient acquisition, especially the phosphorus, but they assist the nitrogen, zinc, copper, magnesium uptake of the plant, of course, and it can help for the soil quality because the glomalin is helped to connect the soil and mycorrhiza. Let's see the importance of it. The root mass increasing when we are using arbuscular mycorrhiza biofertilizer. On this picture, you can see the differences between the mycorrhizal roots and the control roots. You can see that the mycorrhizal roots are much more bigger, and it can more efficient water uptake of this grass, and so it's drought tolerant and also better rest tolerance of with more biomass of roots on the picture. Also, the mycorrhiza can help for increasing the biomass under soil through and cold stress. On the picture in the greenhouse experiments, you can see the differences between the control and mycorrhizal plants, non-mycorrhizal and mycorrhizal plants under control condition, salt condition, drought condition, and cold condition. The biomass increasing is a very big amount, and it's very hard for the plants to be resilient for these stresses. We identified the positive effects on phosphorus uptake of plants. On this picture, you can see the differences between the mycorrhizal and non-mycorrhizal. The black one is non-mycorrhizal, the red one is mycorrhizal plant roots. You can see that under the mycorrhizal plant roots can uptake more phosphorus, so the phosphorus can identified on the leaf and also in other plant parts, more phosphorus. Also, when we are using corn together with different bacterials and arbuscular mycorrhiza, it can be identified bigger root biomass, increasing seed number, and 1,000 kernel weight. On the picture, you can see that, and also bigger crops, so it's mean bigger and more yield when we are using mycorrhiza or bacteria and mycorrhiza together, because in this connection, these special bacteria help for the mycorrhiza to uptake the phosphorus and directly for the plants. In this experiment is a soybean experiments when we use the Bradyrhizobium japonicum, the special rhizobacteria soybean, and together with the arbuscular mycorrhiza product, and we identify the number of nodules on the roots and also the yields, and in the results we identify that together with bacteria and mycorrhiza can give us a higher yield and a better number of nodules, which can give the plants also nitrogens. You can see the picture of these experiments, and maybe you can see the color differences between the control and only one mean with Bradyrhizobium japonicum, and O1 mean Bradyrhizobium japonicum and arbuscular mycorrhiza together, and the color of the plants are changed. So we can identify higher photosynthesis and higher biomass of soybean. We have also another experiments with winter wheat used by mycorrhiza on different fertilizer level. We like to identify the optimal fertilizer level which is used together with mycorrhiza. You can see on the pictures 4 different fertilizer levels, the A, B, C, and D. Under the A fertilizer level, we use phosphorus deficiency, so we never use phosphor. This experiment is more than 20 years old. So always in same plot, we are using same fertilizer level, and we identified that when the phosphorus deficiency is identified, the mycorrhiza, so the red column, you can see the red column, the colonization rate and the yield is higher, significantly higher under the phosphorus deficiency when we are using mycorrhiza biofertilizers. Also, when we see the effects of arbuscular mycorrhiza on wheat yield on different fertilizer levels, the A, B, C, and D levels, which is the D is the highest level of fertilizer, we can identify that if we are using mycorrhiza on the 5.39 tons per hectare of yield are identified lower level of fertilizer than if we are not using. So it's mean that less dosage of chemical fertilizer is enough for the intensive agriculture technology if we are using mycorrhiza. We have experiments with plant protection and mycorrhiza because it's also very important to know that the mycorrhiza help or not help for the plants in agricultural technologies. So we know that everybody knows that the plant has no immune system, so the tolerance and resistance must be appear in every cell in the plant, and it's usually used by the resistance genes. But if there isn't any resistance genes against the pathogen, the induced plant resistance also can help for the plant to be resilient for the diseases. So the early warning, so this induced plant resistance means the early warning, and these arbuscular mycorrhiza fungi can help for this induced plant resistance because they give training session before real danger of the plant. So it's very important to give them training of plants. So when we identify the tomato and the cucumber mosaic virus on the picture, you can see that the mycorrhiza fungi influence the degree of symptoms, and appearance and severity of these symptoms can lower if we are using mycorrhiza fungi. Without that, these cucumber mosaic virus can damage the tomato, and the plant later on died. But together with the mycorrhiza, it's decreased the problem. Strawberry and fusarium also a very big problem in Hungary, and with some farmers we are try to use arbuscular mycorrhiza and check the strawberry plants on the trees on the greenhouse that the fusarium less affect the plants when we are used mycorrhiza as a biofungi against the fusarium. Tomato root nematodes, it's also a very big problem in Hungary and also, as I know, in worldwide. So when we are using arbuscular mycorrhiza against tomato root nematodes, it will be less problem during the growing period. And on this picture, you can see that after the 12 days after the inoculation, the mycorrhiza can help to decrease the root nematodes on the tomato roots, and the yield potential will be higher, and the yield will be much more. The yield quality also will better. Also, there are some papers which can identify that the effects of arbuscular mycorrhiza on the number of second and third stage juveniles and females of nematodes significantly decreased when they used arbuscular mycorrhiza, and the juveniles mean 77 lower in mycorrhiza roots than the non-mycorrhiza roots, so it's absolutely significantly lower, and the females are more than 90% each of lower in mycorrhiza roots, so it's very important results, and it's need to be used. Summary, it's very useful, the mycorrhiza against the nematodes. Of course, the fungal diversity determines the plant diversity. So on this picture, you can see that Van der Heyden and found that the species and diversity of these mycorrhiza can affect the plant biomass, and it's agriculturally it's important to use more than one mycorrhiza species in the biofertilizer products, because together they can affect than if only in one species. Mycorrhizal plants have better nutrient supply, increase the carbohydrate production, so increase pest resistance. The plant invests more energy in shoot development, and as the root system is held by arbuscular mycorrhiza fungi, so that's why the mass and the yield increases, and economically is much more better for the plants and gives us more yield. Of course, there are environmental factors which can affect mycorrhiza development, but I think it's another presentation, because it's a very symbiotic way together with the tillage system, and also the tillage system are renewing of the tillage system worldwide are very important factor for the plants and also for the better agricultural results. Thank you very much for your attention.
Thank you, Dr. Melinda, for a very informative lecture, which the agriculture scientists sitting here and the students would have really enjoyed. Dr. Melinda just told me that in her university she has about 1,000 students in the agriculture domain, and also the university shares its research with the government so that the agriculture produce of the country can improve. I would like to say that we also have a large number of students in agriculture, not only in this university, but other universities, Amity, other universities also. And we also share our research results with the government agencies which look after agriculture. I would like to inform the visitors that the Honorable Founder President who has set up this group of Amity universities, he says that he has put in the spirit of innovation and research. That's why we call all the Amity universities as research and innovation driven universities. And the person who is heading the research and innovation here is Dr. William Silvermurthy, who is the president of the Amity Science and Innovation Foundation. He has been a very senior scientist in the Defense Research and Development Organization and retires as a Director General. And he's respected all over India as well as the world as a very well-known scientist. May I now request him to come and talk about the various science initiatives which Amity University is taking. Dr. William Silvermurthy.
So, I'm so happy to welcome such a distinguished delegation from Hungary. Honorable Founder President, Dr. Ashok K. Chauhanji, who is observing the program right from the very beginning. And he has also been responsible to make the architecture of the whole visit today. And he is the one who has created the whole Amity universe, which is today having 250,000 students, 10,000 faculties, 5,000 PhD scholars, 12 universities, and 18 campuses abroad, 28 schools. So this is a huge universe, Amity universe. So we have great pleasure to welcome such a distinguished delegation. I will start with Professor Thomas Monastery. He is the head Institute of Plant Sciences and Environmental Protection at the University of Szeged. And also we listen to him doing in the field of the agriculture, plant physiology, and also the plant science. We also listen to Dr. Melinda Tal, one of our very distinguished scientists. He is a senior research fellow and an expert in the field of agricultural sciences. We are doing amazing work in mycorrhiza particularly, which is of great interest to Amity as well. We will share with you what we are doing in this domain. And also we are very so grateful to Dr. Diana Daxon, who is coming for the second time. And the whole visit is taking place because of her. Let's give a big clap to Dr. Diana and the distinguished delegation. Dr. Diana, I had the opportunity of interacting with her online last time when she was having lunch in UP Lunch, and because I was away on another important mission. And even today, I am going to Japan today at 8 p.m. So I came only to meet Diana and the whole distinguished delegation. So we are so happy that such a big delegation is here today. And she is the Counselor of Science and Technology in the Embassy of Hungary in India. And she has been here almost for more than 1 year plus. Just in April last year, she took over this responsibility. She has more than 28 years of diplomatic relations, particularly she is a lawyer by profession. She is by her degree. Legislation is a specialization. And also, she has been in various ministries in the Hungary. So she is not only here as an ambassador here, representing Hungary, but also she has been in various ministries. So we can connect with various ministries as well because of her earlier appointments. So such a distinguished delegation is here. We are so delighted. And Amity and the founder president, Dr. Ashok Chauhan, and also the Chancellor, Dr. Atul Chauhan, and all of us have attached a great importance for this visit because we are already doing a lot of collaborations with Hungary. Because Hungary is a country very close to our heart. And we have Dr. Seema Garg, who has just joined. She is doing a big project. Let's give a big clap to her. She is working on photocatalytic reactor. And so she has already developed a system working with industry here as well as there. And so there is another group working. So there is a lot of already initiatives. Students have come here, Hungarian students. Our students and faculty have gone there. And also Rajini Singh has also earlier received a delegation. I think Shakti is going to give a presentation on what all we are doing together. So what I want to tell Dr. Diana is that Hungary is close to our heart, and through you, we will have more such delegation coming and greater expansion of our research and development collaboration, academic collaboration, all this will happen. So I appreciate Dr. Nutan Kaushik. Let's give a big clap to Dr. Nutan Kaushik. So she is here, all of them are here because of her. And the research of our agricultural scientists, all of you. Because of your outstanding research, these distinguished delegations are coming here for collaboration, partnership, and also value addition to their own research. So Nutan Kaushik is the Director General of the Amity Food and Agriculture Foundation, looking after all the agriculture, food related, the research, academics in Amity University and Amity Group. So Nutan Kaushik, she has been with Amity now more than 10 years, and she has been prior to that at TARI, yet another important institute. And I had the opportunity of working with Dr. Nutan when I was at DRDO, because we also have in the defense research, how do you feed soldiers in extreme high operational conditions, like high altitude? Can you cultivate at minus 30 degrees food there? So horticulture produce particularly. Otherwise, to carry 1 kg of tomato from plains to high altitude, the cost is 200 rupees is the freight charges. The cost is maybe 20 rupees, 30 rupees, but to carry that to Leh, it costs about 200 to 300 rupees per the kg. That is why we cultivated food there. So we have a lot of research happening. So I was associated with the hill agriculture, high altitude agriculture earlier when I was with the DRDO. So I'm so happy to listen to both the presentations. I'm sure all of you will be making will be interacting with them after my presentation and also after Dr. Nutan Kaushik presentation. Now to mention about Dr. Thomas Monastery, he is a very senior scientist, and also he is the head of this Institute of Plant Sciences and Environmental Protection at the University of Szeged. And his area, as you have seen from the presentation, is his PhD was in CDMA in sense and anti-sense position. That was the PhD. And then plant breeding, plant protection, agricultural biotechnology, fish farming, all these are his expertise, not only what he presented. So cereals, dry bean, and potato, sweet potato, all these areas he has contributed. His area has been cultivation, nutrition, and conditioning of stress resistance. So that is his focus. So such a distinguished person is here today, and who has made a mark in the field of agriculture, biotechnology. Similarly, Dr. Malinda Thar is a plant scientist with 20 years of experience. Legume production, which is not present over here, the pulses, is one of our areas of research, and also nutrient management and fertilizers, mycorrhiza, which she mentioned about strawberry, and also she mentioned about abiotic stress management, particularly for corn, soybean. And she is also working on how do you build resilience, plant resilience, particularly she showed strawberry, for biotic stress. So not only abiotic stress tolerance, but also on the biotic stress. So such a great scientist. So we are all very happy to have you here. Now coming to India, Hungary. Now we have a very strong diplomatic relations today. And we are still, it is growing, and we are looking at strategic partnership between India as well as Hungary. So Amity would contribute significantly, both in academic and research, in this India-Hungary collaboration and partnership, strategic partnership which is happening now. And SMT cooperation plays a pivotal role, as technology drives the doctrines of a nation, both in war and peace. So that is why we have to jointly develop technologies together. So this type of visit, building partnership and collaboration is very vital for the good of the humanity. And now coming to the founder president, I must say a few words which I would like the delegation to know. Founder president himself is a scientist. He is an academician, he is an entrepreneur, and who has spent about 3 decades in Germany and Europe. So Hungary, he might have visited several times when he was in Germany. So Germany, he was a chemical engineer there, and did his doctorate there, and then he became an entrepreneur, and he was the largest non-European investor in Germany when the reunion took place between East and West Germany. And then when the Chancellor of Germany used to say, I brought political stability to Germany, but then this great Indian, that is Dr. Ashok Chauhan, brought the economic prosperity to Germany. So that is the level of standing our founder president has. And when he returned back, he started investing in 4 pillars. That is research, innovation, philanthropy, education. Education, research, innovation, philanthropy. So on 4 verticals he invested. That is why the whole Amity universe has come in such a short time of 3 decades. We have been able to build this universe, which Dr. Chauhan has built and founded. Steered by chancellors, 2 very dynamic chancellors, Dr. Ashok Chauhan, the president, as well as Dr. Asim Chauhan. They were also educated in Europe and West, North American, and they are the drive engines for Amity Group. And now, it's a very opportune time, Dr. Diana, and also the visiting delegation, I'd like to say, it's an opportune time for India to look as a partner, great partner. Because India is now set a goal, it's a big vision, Viksit Bharat. Viksit Bharat is developed India. We want to be developed India like Hungary, US, or Europe. So we also like to we have aspirational India is looking towards transforming itself as Viksit Bharat or developed India, for which science and technology will play a very, very important role. Academics and education will play a significant role. So that is why we are now looking at gross enrollment ratio today is about 30%. And we want to enhance it to 50% in the next 20 years. So it's a huge task almost to double the gross enrollment ratio. So that's a great opportunity for Hungarian universities and also Indian universities like Amity University to work together to contribute to this growth at gross enrollment ratio. And the middle class economic power is increasing. We are growing at GDP of just 7% GDP growth rate. And our GDP is now reaching $5 trillion. And then every year we'll add $1 trillion to our GDP. So that is the power with which we are moving towards. So this is an opportune time to see how do we work together. The Prime Minister Shri Narendra Modi ji has given a very important slogan, Sabke Saath and Sabke Vikas. That means we want to work with all countries, particularly Hungary and other countries, Sabke Saath and Sabka Vikas. So the Amity has got the same philosophy which founder president has given, that we connect with people, we bring synergy to your envelope, and we also bring outcome from this partnership. We would like to bring the outcome from our partnership. And the National Education Policy 2020 has given all opportunities, flexibility for building this collaboration in a big way. Like, for example, we can have joint degrees, dual degrees, and also campus in campus. And we can have Hungarian campus in Amity Some of your Szeged University can look at the campus in Amity, and similarly, Amity will find a campus in your university. So this is what is the level at which now the flexibility has been given. And so we like to use this opportune time. The window of opportunity is great now. And Amity missions, Mission Connect, Mission Synergy, and Mission Outcome. And in fact, Group Captain Kapil Shukla, who spoke down, he was the Director Outcome. Now he is with the FSTR. So this is a great opportune time. Now I will give you a little glimpse of Amity research and innovation. So this is such a distinguished delegation, Dr. Thomas, Dr. Diana, Dr. Melinda. Welcome to India, welcome to Amity. I'm sure your visit to Amity as well as India will bring very, very fruitful outcome. You will enjoy your stay here. Come more often. We'll be delighted to host you here. So today, to give a glimpse of what we have achieved in the last two and a half decades, we are now 250,000 students, 10,000 faculties in 12 universities in India, 18 campuses abroad like Dubai, Mauritius, Singapore, London, Romania, USA. And we also have about 400 programs, academic programs. We have almost about 800,000 alumni across the globe, including some of them maybe even in Hungary. Just to give a little glimpse of a few campuses, not everything I've put here. This is our Noida campus on your right, on your left extreme. Then we have Amity University, Maharashtra, Mumbai, Amity University, Mohali, Punjab, Hyderabad. So we have like this 12 independent universities. They are not campuses. They are actually independent universities. And then we also have overseas campus like in Dubai, Mauritius, Singapore, London, Romania, USA. We have 2 campuses. One in the Long Island of New York. We have 170 acres. And we have 11 buildings constructed, and we have started a Harrow School there, Amity Harrow School and Amity Global Research Hub. We are now building in Long Island's campus. So we are now top, even in schools. We have 28 schools, including 3 schools. So these are all, they are also doing extremely well in academics and research. Some of these students are filing patents, they have startups, and also they publish papers with journals of repute. So we have right from pre-nursery to postdoctoral level, the whole educational value chain Amity has established. Dr. Ashok Chauhan, the founder, president, and the chancellors, and the madam chairperson who looks after this school, who have established these. Schools we have in overseas as well, like in Amsterdam, a beautiful campus. In Abu Dhabi, it looks like a university. Please do visit. Whenever you are going to those parts, please do visit us. And so these are Sharjah, these are the places where in Kenya we have schools. Then we have also partnered with UK Harrow. This is another very important initiative, that we have a Harrow school. In Bangalore, we have a Harrow School, and we also have one in the Long Island of New York. Beautiful. And the president of Harrow visited just 15 days or 20 days ago in our Amity Harrow in Long Island of New York. He was the one whom I was mentioning. All that what I presented have been founded, built, nurtured, led by this great man, Dr. Ashok Chauhan. I am sure that he will be observing this. He may be interacting with you at some time, some part of the time. Dr. Mrs. Amita Chauhan, she is the first lady of our Amity Education Group, and she looks after all the international schools, and also Mrs. Sapna Chauhan looks after the, and she is PhD in Sanskrit. Dr. Mrs. Amita Chauhan, who is the wife of the founder, she is the chairperson of Amity Schools. He is our chancellor, who is the product of University College of London, Dr. P. Chauhan, and also College of the London School of Economics for the management degree, and also University College of London for engineering degree. So he is the president of the foundation now, and also the chancellor of this particular campus and a few other universities of Amity. He is another chancellor and also the additional president of our foundation. And he says our endeavor is to nurture holistic world citizens. So this is the philosophy of Amity education. We want to develop holistic future global citizens who are endowed with knowledge, including spiritual knowledge, skills including soft skills, good behavior, sanskars, values, resilience to face challenges in life. This is what our faculties, all of them are doing. Let's give clap to our faculties who are doing this great job. So this Dr. Asim Chauhan is a product of Boston Business School, and also he is the additional president of the foundation and chancellor of a few of our Amity University. Now looking at the ranking and accreditation, in the NIRF, even though we are just 21 years young, we began university at the age of 2005, and we are now in 22 in the national the ranking. We are 22 in the country, and the QS and Times Higher Education have rated Amity universities at the top 3% globally. And QS, in some of them we are in top 200. And in employability and international relations, we are in top 100. So that is Amity today, that we are among the elite club of the IIT Delhi, Indian Institute of Science Bangalore, IIM Ahmedabad, and that is the elite group of institutions in which Amity is figuring. And this is QS, and we are in top 3%, and 22 amongst all the NIRF ranking. And then India is number 1, as ranked by some of them in this general category as a private university. We are number 1 in the country, and I'm sure the global universities, I don't think any other organization will have in 18 countries where there are different campuses. And we are also we have NASC accreditation, QA accreditation for quality, as well as for the education and research and innovation. Then we also have been awarded many IPR awards, intellectual property awards, patent awards, because Amity has filed 2,870 patents now, that's the number today. And then we have been awarded the Ministry Government of India, and as you see, some of them being received from CII, Confederation of Indian Industries, Niti Aayog, and MeitY, and many organizations have recognized Amity's contribution to IP ecosystem. ASSOCHAM is yet another organization. So this is a very good index that we also contribute. Our students also win the innovation awards. Like these are Amity International School, Bihar, and they also got the International Property Awards. Atal Tinkering Labs we have in every school for creating innovation and the creativity. Then we were also conferred various awards, which you see here, Founder President Dr. Chauhan receiving from the ministers like Minister of Science and Technology, Dr. Jitendra Singh, and other ministers and governor. Then coming to how research ecosystem, we have two important foundations. Amity Science Technology Innovation Foundation, where I am the president, looking after all Amity universities and institutions for science, technology, innovation. We also have another Amity Foundation for Science, Technology and Innovation Alliances, which builds partnership, research collaboration. Because in international research collaboration, we are in top 100 as per the Times Higher Education. And globalization and internationalization, again, we are in top 100. And we have the specific technology missions. We have in space, we have Amity Space Mission, Amity Quantum Mission, and Amity Artificial Intelligence and Cyber Security Mission. So these are all mission mode, defense technology mission, and we also have health technology. So agriculture and food technology is yet another vertical. So we have built verticals when we grow horizontally. We have also built verticals in specific areas of emerging as well as deep tech. And we are among the India's leading research-driven university. We call it as research-driven because even curriculum design, pedagogy, method of delivery, everything is aimed to promote research and innovation. As you see here, 48,000 research papers published by Amity, 48,000, and our H index is 208, which is again shows the impact of our research. 208 is our Scopus H index. And then we also have more than 500 funded projects, 35 technology transfers we have done to the industry. So we connect with industry, we work with industry, translate the scientific leads into a deliverable, either a product, technology, or a process reaching out to the people. We have excellent laboratories, and some of them I have mentioned NMR 400 megahertz, scanning electron microscope. We have 4 3D printers, we have bioprinter, and also fully automated design labs, as well as histopathology, stem cell, tissue engineering, and agricultural labs. These are the labs will be looking like this. Some of them, please do visit when you are here. And we have built wonderful facilities, even BSL 2 plus facility we have. When we deal with pathogens, so we can also handle BSL 2 plus. And then we have advanced instrumentation, atomic absorption spectrophotometer, and also the fax machine for cell studies, and a number of them. Then we also, US Department of Defense, through DITRA, Defense Threat Reduction Agency, invested $1.2 million in Amity and built a nuclear security lab. Now, how do you build capacity in terms of securing the nuclear assets? So for detection, handheld, standalone, standoff, detection for alpha, beta, gamma, and neutron particles, how do you detect them so that the nuclear assets in the civilian sector is secured properly? So we have such a huge investment, and we are building capacity like NDRF, SDRF, and also other security agencies are being trained in our facility, and we also do research using this facility. Then we also have AI Center. We have a 10 petaflop computing capability today in this campus, 10 petaflop. We have the NVIDIA A100 system, and now this 10 petaflop computing capability we are expanding or augmenting to 60 petaflop. So we are using it for agriculture, defense, space, as well as in the energy sector. And we are educating every student in the field of artificial intelligence, because it is almost all pervasive. Even in governance, you can use this artificial intelligence. And then this is we are also encouraging startups from our faculties. Our faculties can also teach, do research, and can become a director of a company. So we have given this dispensation, and they don't have to take a sabbatical and leave, and then they can be simultaneously devoting time for their own startups and being an entrepreneur, taking their own technology. One such technology is that we have developed a polymer sponge, nano polymer sponge, to quench, clean the oil spillage in ocean as well as in all the oil installations. So this is a very important startup, which is now becoming very connected with so many industries, and so many organizations like defense, and also ONGC, others, they are now showing interest in the technology. These are the verticals: data science and AI, nanotechnology, robotics, astrobiology. I would be happy to inform you that we were the first one to send the first biological payload to space from India, then from Amity. That was a callus of a plant which is spinach, which we have put it and then looked at 45 days in space, that how does it behave in terms of plant physiology. We were looking at rooting, shooting, and also callus formation, biomass formation, plant physiology. But if you have to cultivate agriculture in space, that how do you do that? What are the challenges for an agricultural scientist? So we have astrobiology, and in astrobiology we were also doing work on the Mars analog station. We have created in Ladakh a Mars analog station which can simulate the conditions of Mars, including terrain, atmospheric conditions, the extremophiles, as well as the psychological stress of isolation, monotony, separation from family, fear of the unknown, sensory deprivation. You can create that. So we have, Amity has a Mars analog station. So astrobiology is one vertical. Cyber security, we are building a center of excellence in cyber security, knowing the importance of cyberspace protection. So we are now doing capacity building as well as research in this field. Then we are going to establish a range, cyber range here. So that you can simulate red team, blue team, how do you attack, how do you respond. So we have such kind of cyber security is another focus. Then space science, I will show you some of the research. Spintronics, artificial intelligence, optics and electro-optics. Then sensors, food and agriculture, strong vertical, bioenergy and biofuels, computational biology, cancer biology, translational research, molecular medicine and stem cell research, genome engineering, neuroscience, virology and immunology, quick chemistry, drug development, and Indian systems of medicine, and motion and atmospheric science. So the whole canvas is huge, These are the verticals in our group we are building. Now, some of the innovations, just I will give a little glimpse. This is the AI platform which we are already developed and ready for use by the security forces. You can identify an individual, even if the face is covered and head is covered, and you can identify using the skeletal configuration, gestures, and the gait of the person. Precisely so, this is the person. So this is very valuable. We are using that for maritime security. Maritime security, we have Amity Dubai, we have a ground station for space. So all the satellite data which keeps flowing, and this year we will be launching our 3CubeSat, the 3Cube system to space for atmospheric monitoring. And this is the plant module which is sent to space through ISRO. We have an Amity quantum mission. Our Dr. Atul Chauhan, the Chancellor, has launched an Amity quantum mission where we are looking at quantum computing, quantum sensing, quantum communication, quantum materials, and meteorology. So these four verticals, we are running MTech quantum technology from this year onwards. We have started a minor. So these are the areas, these are some deep tech areas where we will also be doing research. We are doing quantum key distribution for the armed forces. So secure communication, we are working with the DRDO. Similarly, green hydrogen, green energy solution is a very strong vertical here because India will become carbon neutral by 2070. So how do you reduce the footprints, carbon footprints? And so one of the missions is green hydrogen mission, Amity green hydrogen mission, which the founder president himself steers this mission where we are looking at cost-effective production of hydrogen, green hydrogen, and then we are looking at storage, transportation, and utility as well. Then this is very important for you, which that Newton will also be talking about it. You talked about mycorrhiza. Mycorrhiza, we have already working on root endophyte, which is a plant growth promoter, which is Piriforma spora indica. And now we have developed a consortium, which Dr. Amit will be talking about this. Amit Karkwal will talk about this. And biofertilizer plant is a bioregulator and a bioherbicide. So it works on cereal, pulses, horticulture produce, sugarcane, medicinal plants. You name anything, this particular plant works on that. It is known as Piriforma spora indica. We obtained from the Thar deserts of Rajasthan, but it works in sub-zero temperatures in Ladakh. So this is the unique feature of this Piriforma spora indica. And then Acetobacter and Tellurium is this, and which he is working as a consortium now, not just only one root endophyte, but we are looking at a consortium of microbes, which can increase both abiotic stress, like low water availability, which you showed using mycorrhiza, and also the salinity of the soil, alkalinity of the soil, and so this is what this fungus does. It's a fungus. Then this is the combination which Amit Kirkwal he has prepared, a very novel consortium, which is also he is looking at the biodiversity. Also, the endophytic activity of Telluromyces is the one which he is looking at, and he will talk about this as a plant growth promoting agent. And these products have reached the farmers, not that they are in the experimental stage. We are transferred to 9 industries. These two technologies have been taken by 9 industries, including one in Taiwan, the Sinan Corporation, and also one in Canada, the Premier Tech Canada. We are now looking at the further transferring to other agencies. And what we have done to double the income of the farmers, we have made a new approach. On the agricultural field, you cultivate the crop on the ground and then erect the poles, put the photovoltaic, and put the water sprinkler on the top of them to remove the dust and increase the efficiency of photovoltaic system. And the transpiration of the plants acts like a heat sink, taking away the heat, so that the heat of the bottom of the photovoltaic cell will be reduced, thereby efficiency goes up. So we have made a demonstration plant here. We are replicating this solar farming in many of this. Maybe you can look at this through one of the collaboration for your agricultural field as well. Then we have a very strong post-harvest technology center here, because our country is Now today temperature is 47 degrees, it might reach 47. So at this temperature, all perishable fruits, vegetables, they all perish fast. So how do you manage the agricultural produce in a post-harvest stage? So we have three technologies which we have given. Where there is no electricity, you can use a zero energy cooling chamber. You make a double wall brick structure, put the husk over it, put the water sprinkler on the top of this. So the water keeps dripping through the husk, and then it reduces the air temperature between the two layers of these bricks, and then thereby the temperature comes down by 8 to 10 degrees, which is sufficient enough to give a zero energy cooling chamber. Then we also have sensor development, AMyCool, which can optimize the electricity consumption wherever the electricity is available, and using the ambient temperature outside and the desired temperature inside the chamber. So the differential, if you look at, and optimize the electricity consumption. Third one is a reversible blocking of ethylene receptors, which is MCP1, which we have tried in papaya, mango, banana. So the shelf life improves up to 45 days. You can preserve it using this reversible ethylene blocking. So this kind of post-harvest is a very strong group in Amity. So this is what I have shown the data, that you have mangoes, which is now the season is for mangoes. So you see that it goes up to 45 days. I think this is packaging, green packaging. Again, we do biomass, you throw up like cabbage, you have a lot of biomass. How do you use it to convert them into a packaging material? And we are also developing a vegetarian capsule from a biological system, biomass. And then these are edible coatings, which I think she will be showing. This is to increase the shelf life, particularly fruits and vegetables. Particularly fruits, can you cover it with the biofilm, biocoatings? So this research has also happened in our Amity in the biotechnology. And these are circular economy is another area where Amity is working. We don't want to throw up. How do we reuse this, recycle it? So we are doing, like Nitin Kaushik himself is doing a great work on fish processing industry. When the water is thrown out as an effluent, how do you collect the wealth from it, like proteins, amino acids, fatty acids, and also beta-carotene, vitamin A. All this can be taken out, salvaged from the effluent coming from the fish processing industry. So circular economy is very important, wealth from waste. I will just go through now quickly. We have a very strong food technology group. We look at all food preservation, food processing, biopreservatives, and also retard process technologies. And we make all this for the food processing. These are some of the foods which I have. Okay, sorry. So we have value-added products, not only post-harvest management. We convert them into value-added products. We are even developing a space food, working with CFTRI, the Central Food Technology Research Institute. Mushroom value-added products, again, our food technology group is developing. These are the empowerment of the farm women. We are training people through extension so that all this technology gets transferred to the people who will be manufacturing it. By simple food processing industry, like tomato, you can convert them into a beverage, puree, paste, pickle. So we have developed such simple food technology, which the women's self-help group at villages can use it. Like millets, ragi, we have again brought some food. So millet mission, we are also working on millets. These are some of the missions the food technology department has for various preservation, including the moon. These are extension activities. So I have our extension specialist here, who will be looking at taking the technology and learning from the lab to land to the farmers. So we run training programs so that the knowledge is not restricted to lab, it reaches the actual user who is the farmer. And this is the Center for Translational Research. We have a Translational Research Center which is working on tuberculosis. So these are some of the technologies transferred to industry. 35 technologies have been transferred. We have an incubator which you will be visiting today. It has incubated more than 1,100 the startups and ventures, and also 75 investments facilitated. And we have our own Amity Venture Capital, which we also give taking equities, which is seen by Dr. Asim Chauhan, overseen by Dr. Asim Chauhan. So now finally I'll conclude with this message from Honorable Founder President Dr. Ashok Chauhan to quote, his dream is to become India a knowledge superpower by 2030, where Amity will play, all of us will play with you as partners. We will play a very important role in the transformation of India as a knowledge superpower and maybe also general superpower. So his quote, I quote Dr. Ashok Chauhan, the founder president, and I can say with pride that my scientists, my researchers move with this confidence that they will do wonders and will add to the growth of the nation, and this will help making India not only a knowledge superpower, but also a general superpower. Thank you, Founder President, and thank you so much to every one of you.
Thank you so much, Dr. Subramurthy, for bringing out the research and innovation flavor of Amity universities, and especially in the agriculture domain. Now there will be, before we have an open house, where we will introduce all our scientists and faculty members who are here, and then they can discuss various points which they have with the visitors. There are two small presentations and a small talk, for which I would request Dr. Shefali Kashyap, Senior Additional Director of FSTIA, to kindly coordinate.
Thank you so much. A warm welcome to our guest, Professor Thomas, Dr. Melinda, and our friend, Dr. Diana. I am Dr. Shefali, Senior Director, Amity Foundation for Science, Technology, Innovation Alliances and SNT War Room. Our War Room is directly governed by our founder president, Dr. Ashok Chauhan, and I am one of his proud SNT warriors. After having detailed presentation from Dr. Subramurthy, now it's time to discuss and share a brief highlight on the Amity's collaborative work with Hungarian partners. Deliberating on this, may I request Dr. Nutan Kaushik Madam, our Director General, Amity Foundation and Agriculture Foundation. She will emphasize on the agriculture and the possible areas of collaboration. Thank you so much. Over to you, ma'am. Very good afternoon to all of you. I'll just give a presentation about what we are doing in the field of agriculture and allied area. Just to give you, I want to give the glimpses of the activities which are doing, so that so that we can develop the ideas of collaboration for future collaborations. Thanks, the director for this. 100. Okay. Thank you. So we are very proud to say that in India we have a ranking system called the NIRF ranking, which ranks all the universities in different sectors based on their capability, research, academics, and everything. So we are very proud that among all the private and government universities, Amity University is ranked number 15 in NIRF ranking in 2025. We applied for the very first time and we were ranked very high. And we were number 3 in research and innovation in agriculture and allied sector. So this is our honorable founder person, Dr. Ashok Chauhan, who found this Amity Food and Agriculture Foundation because agriculture and food are very close to his heart. So since the inception of the university, the Amity is the only private university which has agriculture as a core sector not only in the academics but also in research. Research because he established various institutions and research centers focused, and then with that, and then later on he created Amity Food and Agriculture Foundation to promote the research. So the objective of this foundation is to conduct research leading to excellence, dissemination of innovative technology, imparting new education in agriculture-related fields, and developing globally competitive human resources. And these are just to give you a glimpse that what are the various centers and institutes which are dedicatedly working in this area. So we are Amity Institute of Horticulture Studies and Research. We have Amity Institute of Organic Agriculture. Dr. Doily was sitting here. She is the director of AUUP. The Amity International Center for Post-Harvest Technology and Cold Chain Management. Amity Center for Biocontrol and Plant Disease Management. Dr. Mehta Singh is sitting here. And the Amity Center for Exchange and Services, which is again headed by being looked after by Dr. Mehta Singh. The Amity Center for Soil Science, the Amity Institute of Food Technology, Center for Agriculture Biotechnology, and Amity Institute of Herbal Research and Studies. So these are the various centers, and these are the various thematic areas which we are working on. So improving agriculture productivity by tackling the biotech and abiotic stresses, and post-harvest management and value addition, improving nutritional status of the soil and crops and food, and utilization of the food processing industry and agri-farm waste, medicine and aromatic plants, vertical farming, integrated farming, climate-smart agriculture, smart and precision farming, and training and outreach. So these are our focus area. And in academics, we offer these programs. We offer BSc Honours Agriculture, MBA Agriculture and Food Business, MSc Agriculture Agronomy, MSc Horticulture Post-Harvest Management, and PhD in Agriculture and PhD in Horticulture. And these are the 4 pillars of excellence in academics. So we give a lot of international exposure to our students, and I hope this particular exercise which you are doing is another step in this direction, that our students can come, go and visit your institution. So, and then well-met program structure. So we keep on improvising the syllabus every year, depending upon the industrial requirement, depending upon the societal requirement. So we improve the syllabus every year, and when strong industry exposure to the students, we take our students to different industry units, we invite industries here, and then state-of-the-art laboratory facility for doing the high-quality research. And this is some of the lab infrastructure which we have here: HPLC, GCMS, atomic absorption, we have coaching, we have plant-growth children, food processing pilot plant, fermentation facility, and the lab cooling chamber. And this is the agrivoltaic. Dr. Silvamurthy sir has already presented about it. This is our organic farm, just sitting close to this particular building at the corner. If you have time, you can visit this farm also. And so we have got the projects from international national funding agencies. Like recently, we have completed one, in the EU project. There were 6 partners in this one, including Germany and Spain and the Norway. From the EU, and from India, we were the University of this plan were the partner. Then from Asia Pacific, Japan, we got this climate smart agriculture practices. And then from RC and Norway, we have done these fish projects that utilizing the fish waste. So we have done 3 projects from RCN support. One is on the fish waste utilization, another on the oil waste utilization, and so on. So likewise, and we have done from CBBC to Russia. And we have signed an international collaboration. These are whatever we are reflecting here are just for the agriculture and life sector. Amity University has more MOUs. They have more than 200 MOUs signed. But these MOUs are dedicated for the agriculture and agri sector. And for the very well, another time when I present, I would like to add the logo of the university in this particular slide. Okay. So we have at CSIC Spain. CSIC is just like the CSI in India. They say that network of institutions are like 150 institutions across Spain. You can collaborate with any of these institute, just like the Amity Institute is a group of institutions. Simpak, Ocean, NTNU, UW, Australia, Wageningen University, which is one of the top most university in agriculture, Ohio State University, with Professor Vandal, which I was mentioning, we have an MOU, and with the Strathclyde Glasgow, we have an MOU. So, and these are some of our honorary doctorate and professors, and we want to wish to add more here from Hungary. Okay, so Professor Ratan, who is a World Food Prize laureate, equivalent to the Nobel Prize in Agriculture, and then Professor Peter Koch from Germany, Professor Kadambok Siddiqui from Australia, then Professor Vida Srivastava of the USA, then Dr. Jadavali from Tunisia, and then Professor Dr. Purvi Datta from Bill and Melinda Gates Foundation. And we publish in good, we have published in good journals with the cumulative impact factor of more than 400. These are some of the journals, some of the publication. These are some of the awards which we have got. About 3 of our scientists are in top 2% scientists of the Stanford in the current year, 2025. Then we were given the Academic Excellence Award in Agriculture for Amity University was awarded. And this is the Bioagri Award. Then these are some of the glimpses of our projects. We have completed more than 40 projects in this area, including 10 international projects. So this out of 40, 10 are, it means 25% of our projects are international projects. So there is strong presence, international presence in agriculture field. And with the total value is more than 18 crores, which is 1.8 million Indian rupees. Many technologies have been developed. Some of them has already been highlighted by Dr. Sivanati. I will give you more details later on. And we have published more than 600 papers and more than 50 patents. 24 patents are granted. It's not that we have only 5 patents. The patents are also granted. And we have done 6 technology transfer. And then 15 active MOUs and more than 50 PhD scholars are working, currently working. And more than 300 master's students are currently working in different, tackling different areas. And then more than 50 training programs were conducted for farmers. We directly connect with the farmers. So we work in the high-end laboratory and also communicate whatever we do to the farming community. We believe in transferring the knowledge and the technologies to the farmer, whether it is developed by us or it is developed by someone else. We take it to the farmer and transfer it directly. And then we have entered, these are some of the biopesticide, biopetrol, PGP and biostimulants developed at Amity University. So Dr. Silva Botis already talked about the Rupanic, which is a Piriforma spora indica. Dr. Amit Kharagwal is saying 2 of the technology, HMB binding and Rupanic are developed by Amit Kharagwal. He's sitting over here. And these MNCs based bioformulation has recently been transferred to, all have been transferred to the industry, all these 3 technology. And then PCMS has been developed by me in the Tunisia project. And this is a PGPR and the antifungal and biostimulant all together in one person. And we have some of the other technologies, like you can see this P-solubilizing biofertilizer, and then from the food product, fish waste product, and then the development of this Rupanic. I think these have been already presented by Dr. Sunanda sir. This is our Indonesia project in which the DCMS's project was developed. And this is the Revalue project where we developed this, converted the fish, this is the Indigo project, where we converted the fish waste into the value-added product. And we also developed the technology for removal of the proteins from the wash water, the industrial wastewater, so that the water can be discharged safely in the sea, back to the sea, or can be used for the fertilization purposes. And in the micronutrient deficiency, we are working with the agronomic biofortification. Some people are doing the biofortification through the breeding, some are doing biofortification of the food, but this is the biofortification by spraying the micronutrients on the foliar spray of the macronutrients. And we found that foliar spray is much better than the soil application. So this way, we have developed a microbial managing and which can be utilized by the plant efficiently through the foliar spray. And this technology has been demonstrated in more than 500 fields at farmers level. This is a research network project on linseed and oilseed crop, and in India we have more than 2,500 germplasm of linseed. So here the objective was to characterize this germplasm and identify which are the suitable ones for having the high libellinic acid content or the high fiber content or the dot tolerance and so on. And Amity is involved in one of the sub-project, which is the biochemical profiling of the linseed germplasm. So in this component, we have analyzed all the germplasm, more than 2,500 germplasm, for fatty acid composition, for lignin content, and for this amino acid profiling. And this paper has been published in good papers, and all things have been published in. So these are the papers which have been published based on this characterization of this linseed germplasm. I'll share this PPT with you so that you can use it. And then this is one of the, we have developed one technique by which you can differentiate the raw material based on the HPLC fingerprinting. Normally people do the DNA fingerprinting. But if you have the extract, you cannot do the DNA fingerprinting. So in medicinal plants, when the extract is coming, you don't know whether the extract is coming from one species or another species. So by this method, you can differentiate the extract also. You can pinpoint the species and the plant part from where the extract is coming. And this is the Botania somnifera, is a medicinal plant. So we characterize this bioprospected about 165 germplasm, and we profiled them based on the HPLC profile in two chemotypes, chemotype 1 and the mixture of the chemotype 1 and the chemotype 2. And we also saw that content of the godaferin content, we also developed the harvesting time for the godaferin. So which is the best time when we can get the maximum godaferin. We found that before the maturity is the best time to harvest, get the maximum godaferin content. And this is the Tylophora indica. This was an endogenous project which we did. And where because Tylophora indica roots are basically used by the Ayurvedic plants. But the leaves are not used. So our objective was, can we give some value to the leaves also? So we isolated some compound from the leaves, and that which gives a very good insecticidal activity. So we developed a biopesticide from the leaves. While the roots are being used by the Ayurvedic plant, the other raw material which is left over, the leaf is heavier given the value in terms of the biopesticide. And so we did this LCMS-based profiling to isolate this one. Then another project was the Indo-French project, where we characterize the saponins, because chlorophyton is a plant which is being used in traditional India, but its chemistry was not known before we started this project. So we isolated very vulnerable site from this one and characterized them, and also did the HPLC fingerprinting of this one, all the chlorophyton species. This is the variability in the berberin content in berberis. I do not know whether you have the berberis plant, some species of the berberis in Hungary or not, but we found lot of variability, 1.6% to the 0.0099%. So you can identify the plants which are having the high berberin and use them for this. Then another project we did was for the sea buckthorn. Sea buckthorn is very popular. I think the oil is very popular. It also contains lycopene, beta-carotene. It also contains ascorbic acid. So we identify the physiological stage. There are 4 physiological stages, and we characterize all the 3 components in all the stages. So if you want, so depending upon your requirement, you can identify the stage at which it has to be harvested. The berries has to be harvested. So we found that in phase 1, the arbituric acid is very high, whereas if you want the beta-carotene, then stage 3 is the best stage. So according to your requirement, you can harvest the berries according to the physiological growth of the plant. So, and then there was another project on the moon, Again, there was more than 3,000 new seeds were collected across India, and I was a chemical coordinator for doing the characterization of all the seeds which happened for azathioprine content, oil content, and fatty acid composition. And we found that azathioprine content was 0.02 to 1.6%. The problem with the bean was that people used to say, some people used to say it works, some people used to say it does not work. And the reason was the variability. If you are having low ASA content BB, it will not work. If you are having high, it will work. So variability characterization is very important. So this is again, we did the similarity analysis and all that. And we will take, this is the APM project where we identified various practices for the climate smart practices. So we had, this was in Bangladesh, Afghanistan, and Australia were the partners in this particular project. And we trained the farmers on this extraction services. We also own farmers producer organization, 7 FPOs in different companies. FPOs are just like the farmers producer company. So we help the farmers in formulating in registering their companies and doing the business. So this is a government of India based project which we are doing. And in the post-harvest, Dr. Senior Professor has already provided. But another thing which I want to highlight, we are also working with the solar-powered cold room channels. Solar-powered cold rooms are one which are being used by the pack houses before the export. So this is the ZECC. This is for the marginal farmers. So they can store their produce without energy very easily. And one of it has already been demonstrated in Afghanistan. So this is one of the PhD scholars from Afghanistan who established this one, and this was found very popular among in the market. He established in the market actually. So those people who are buying it before, if the produce is not sold, they used to keep it in the ZECC storage till the time it is sold in the market. And this is delayed ripening. We worked on the delayed ripening. MCP1 was the compound which we tested, and we found this compound can delay the ripening of the. We tested this compound on various food crops like mango and banana and papaya and so on. And we give the value addition training to the farmers, like suppose the farmers are producing the surplus, how to do the value addition and then market it directly. And we also have the agri-entrepreneurship cell. There's our students. We register in this agri-entrepreneurship cell, and then there are various. These are some of the startups which has come out after this agri-entrepreneurship cell. And these are some futuristic this. We are now working towards agriculture 5.0, where we are working on precision agriculture, robotics, data insights, and hydroponics, blockchain, the value chain, and then vertical farming, and so on. These are the different verticals, and climate resilient smart agriculture, labels, metagenomics, and proteomics, developing the varieties, identify the plants and the traits, and so on, and functional foods, alternative sources of proteins. After this war, everybody is talking about the oils and proteins because we want to become the self-sufficient in this particular area. So these are some of the areas where we could collaborate put together. Organic agriculture, we have dedicated institute on organic agriculture. This is one of the area. Now when we are facing the shortage of the fertilizer, everybody is talking how to sustain without chemicals. And organic becomes very important. So we have some findings and which we can share, and we could together work on this one idea. Then validation of food processing industry, we will continue working with plant health, soil health, and then training and consultancies. So these are some of the areas, and this quote has already been given by Srinamurthy sir. So thank you so much. I want your picture next time, picture with all of you here. So we'll keep it there. Thank you. Thank you so much, ma'am, for sharing your outcome-based research work in the food and agricultural sciences. A big round of applause to madam. And seeing the excitement on our those faces after learning about your research work, ma'am. And it is my duty to proudly mention that madam comes on the top 75 women in chemistry by RSC, DBT Crest Fellow 2012, DAD Research Ambassador 2010, Fellow of Royal Government of Netherlands 2005, INSA Young Scientist Award, and then also she comes on the top 2% scientists of the world. Thank you so much, ma'am. You are asset for Amity University. Taking our deliberations forward, now may I request Dr. Seema Garg, who is great scientist and associate professor of Amity Institute of Applied Sciences. Professor, yes, I'm so sorry. And she works in materials, water treatment, and energy. And she is also the great contributor in our Amity Hydrogen Mission. Over to you, ma'am. Thank you so much. Thank you, Dr. Shefari. Honorable Founder President, sir, Chancellor, sir, Vice Chancellor, ma'am, respected President Astif, Selvamurthy, sir. He is not only president but our mentor also, and always supports all the faculty members. So, sir, we are really grateful for your support as mentor. And you were Mr. Pandey. So we all welcome our delegates. Dr. Diana, we have already met earlier when you visited Amity, and Professor Tamas and Dr. Melinda. Welcome to Amity. I'm Professor Seema Garg. Recently, I have been shifted to academics as Deputy Director Academics. I'm Professor at Chemistry Department. And my collaboration from Hungary was started in 2016 with Professor Clara Harnadi. She was professor there at that time. Then in 2022 or something, she shifted to University of Miskolc. So with both university, we have MOUs also. Thank you. Miskolc, yeah. So first of all, I'm really thankful for giving me opportunity to present my research collaboration with Hungary. I will say India, like I have visited 3 times Hungary, and I felt always, I don't know which connection is like with Hungary, because I always feel these are my people kind of with Hungarian. Every time they are very sweet, and they support everything, whatever in research we require, and with collaborative effect, actually we have done a lot of work. So I'll show here, you can see that the 2 projects I have completed with Hungary. These are the complete projects, not only the visit project, and that I got the JRF, amount for consumable, contingency, and visit. And they visited also 3 times in the projects. So first project I got on 27 October 2016 with Professor Clara Harnadi as PI, and from here I was PI. And the second project, it was sanctioned in 2021 with IIT Delhi people. So these projects, you can see 9+ years of we have active collaboration, 30+ we have completed joint publications, and 12+ students have already got taken benefit from these projects. So beyond research publication and projects, you can see the projects gave opportunity to students also to pursue their PhD, postdoctoral, and project work at University of Szeged, like major project and internship, with full stipend support. So these are, and the collaborators, you can see, I am Professor Seema Garg, Professor Sameer Sapra, Professor Ingole was also associated with us from IIT Delhi, Professor here from here, Dr. Sumant also is in our team, and from Hungary, Professor Clara Harnadi, Professor Zoltan Ponya, Vice Rector, University of Szeged, and Dr. Andras Sapi. So the collaboration, and we are also doing the work. Like few students have work under both scientists from Amity University and Hungary. So MOU has been established with University of Szeged and University of Miskolc. So these are the PhD students. These all 4 students completed PhD under like my guidance and the guidance of University of Szeged and University of Miskolc. Like Miss Nikita Sharma, she was alumni of 2012 to 14 batch of MSc. She joined PhD under Professor Clara Harnadi and myself. with full stipend, and now she's also doing postdoc there. And Mohit Yadav, he completed his PhD from here under my guidance and Professor Clara, and did his postdoc. So similarly, Harshita, Isha also got postdoc there, but she couldn't go because of some family reasons. But she got the opportunity, but she did PhD under both scientists. So these are the students who went for summer internship first, then they went for the major project for 6 months. So these are the girls of our 2018-20 batch. These are all our students of applied sciences because I am faculty member of applied sciences. So I have quoted the pics of these. Nishat Khan, she also went batch of 21-23 went there for her major project. And apart from these, pharmacy students also visited Hungary, and one student, Yamini Kanti, got fellowship over there under Stipendium Hungaricum for her PhD, and then she joined their postdoc fellowship. So I've not mentioned that. These are the joint publication. You can see 30 plus joint publication. 11 are the highest. This is the recent publication, transition metal Rodin UCS applied energy material impact factor 6 and 5.5, 11 impact factor 9.5, 6.6. And these type of publications we have more than 30. And you can see the Springer book one. And we have 5 patents also because of this collaboration. So these are the patents and mutual visits, award and recognition. I visited 3 times consecutive years, 2017, 18, 19 for 15 days every time. And I visited their labs and some training on instruments, joint research discussions and lectures were delivered by us in their conducting workshops. And similarly, they also visited here 3 years consecutively, 17, 18, 19. And I got certificate of appreciation from University of Szeged in 2019. And when Clara Harnadi visited in 2019, we conferred honorary professorship to her in 2019. So these are the achievements we got from this Kozono. Thank you. Thank you, sir. Yes, surely, sir. Actually, it could not be, it was possible because of sir's guidance always, because that time I needed guidance, and you know that I was so bad that day, and sir motivated me to just submit the project with full positivity and all. Thank you so much, sir. Thank you so much. You always also motivated. Thank you so much, Dr. Seema Garg. You have a remarkable knack of building collaborations, not only with national universities, but also with international universities. And your scholars are doing a great job under your able leadership. Now, may we request Professor Meghna, Professor of Amity International Business School. She is Professor in Economics and Strategy Innovations. And Professor Jimmy Deka, Amity International Business School. She is working in financial technology. And they will discuss more on Erasmus project and some projects with Hungarian counterparts. Over to you, ma'am, please. Thank you, Dr. Shivani. Thank you so much. Welcome, Dr. Diamond. The more we talk about Hungary, Hungary is very close to our heart because we'll be working with Indian University. Welcome, Dr. Thomas and Dr. Nandal. Your work is outstanding. The more you were speaking about your research, more inspired and motivated we were feeling when we were going through. So while I might not be an agriculture scientist, but there's a commonality. When you talk about seed, you may be teaching farmers or helping farmers to manage seed. My research kind of contributes to manage funds. So my research area is microfinance. So somehow we are connected with the farmer. Farmer is a commonality between you and our research. Yes. We'll be talking about me and my colleague, Dr. Jimmy Deka.
under the guidance of Group Vice Chancellor Dr. Gurinder Singh, we submitted this project. And with my gratitude to our leadership, Honorable Founder President, Honorable Chancellor, Honorable Group Vice Chancellor, Honorable Vice Chancellor, Ma'am, and all the people sitting on the board, with my gratitude, I want to share in whatever small little capacity we have contributed to with the partners of Hungary. So the relationship started with just paper writing or paper presentation. You'll be very happy to know we at Amity, we were the first university who got approval to do IEEE conference across globe. So we have been doing in US, we have been doing in UK, we have been doing in Singapore, Dubai, Tashkent, so on and so forth. So from there, the relationship started with Etoile Laurent University. ELT, as you know, and Dr. Diana, I'm sure you have a reason to feel proud because that is your university as well. So during English, we also invited Lorente Cordos, who is the Economics and Commercial Counselor at the Embassy of Hungary, and he categorically mentioned he mentioned for himself that he was very proud because he was the alumnus of this university, and the officials from that universities were there present at that very time. So the relationship started in 2023 when we exchanged our paper. They came to present their paper with us. But as per the mandate given to us by our Honorable Founder President, that we are the persuaders of outcome. So we initially started relationship 23, it couldn't work out, but 24 we were able to do that because we persuaded it and we were continuously working on the outcome. So 2024 it materialized. 2025, 2026 we got the first approval of Erasmus product, which was a mobility agreement, mobility of higher education for staff and student mobility agreement. So these were the members who were part of this mobility agreement. And as part of the mobility agreement, we visited Hungary and they visited India. When we visited Hungary, of course, we were hosted by Etoile Laurent University, but I'll be talking about other universities also with whom we have started our projects with. This was the first funding, which is part of the faculty mobility and student mobility. This is what we are doing currently. We are in this space, and three faculty members from our side will be visiting ELTE, as we call it, and 4 faculty members from Etoile Laurent have already visited us in the month of February during our mega event, which was in Bush. After this agreement, it was for the teaching and training mobility, which is further extended to teaching, training, and research mobility. As part of outcome of this particular initiative, we are applying for joint funding project under Hungary in the funding projects. This is our next year. Fortunately, we have got approval from Erasmus Plus Hungarian team to continue this mobility for next year as well. And you'll be very happy to know the space that they have created. They have expanded the people who will be visiting. Instead of 3 faculty members, there will be 4 faculty members who will be visiting Etoile Laurent University, Hungary. And likewise, therefore, faculty members will be visiting us. Student mobility. You'll be very happy to know that 2 of our students are already selected. They are landing there in August to January. They'll be doing the full semester there as part of the Erasmus project. And of course, need us to mention that it is 100% funded project. So they will be staying inside the campus and everything has been done. They just need to go there in the month of August. Next year, we have also expanded this mobility program for 2 of our PhD scholars. So not only the master's and undergrad student, even to PhD scholars will be given this opportunity to travel to Hungary and be there, take their mentorship, take their guidance. No, they are doing virtually, they are there, they are mentoring our student, but this time the student will be physically going to Hungary ELT and taking the advice of their guides there. So this was for the next year, this has started. This is the small thoughts from which we started our Erasmus Plus Mobility program. Of course, mobility is there, research outcome is there. Next year, 2027, Amity and ELT together, they are doing one IEEE session in US conference at New York. So the approval is IEEE Long Island chapter, where we very proudly say we have our own campus there, 170-acre campus there. For 2027, ELT will be part of our conference and they will be having a separate session there. These are 2 of the pictures which we captured there, and we always say when we talk about Hungary, we feel some kind of belongingness because we have been working together. You can see the team of Erasmus, the classroom sessions, the faculty, and
So these are a few of the moments that we captured when we were there at Hungary. You can see that, the classroom sessions, and at the bottom right, you can see the team of Erasmus Plus who were there. In return, we also were happy to welcome team of ELTE. Dr. Raina, you can see Levente Gados at the center, and he was here. He particularly wanted to be with the team because the team was coming here. And very, very happy to have them here, and we are taking our relationship forward. Thank you so very much.
Thank you, Dr. Meghnad.
Thank you so much.
So we have listened to a few presentations, but a lot of things to be told by our scientists. But I have picked up among the people present some 4 people to add on. I will now start with Dr. Amit Gargwal, who is in the Amity Institute of Microbial Technology, and he is the architect of mycorrhiza, as well as the biofertilizers and Puriformaspora indica, HNB9, all that significant contributions, field studies. So wonderful scientists we have. Let's give a big clap to Dr. Amit Gargwal. Over to you. Thank you very much. Just 2 minutes or 3 minutes maximum.
Thank you, sir. So, good afternoon to the Hungarian guests. I am Dr. Amit Kharkwal, Director and Professor at Amity Institute of Microbial Technology. So, sir has already shown you, we work in the area of plant microbe and microbe interaction for sustainable agriculture. And our cornerstone technology, that is Rootonic, based on Piriformospora indica, developed by late Professor Dr. Ajit Verma, who is also a very legendary scientist, was also a very legendary scientist of the mycorrhiza technology. So this Piriformospora indica or Serendipita indica is a mycorrhiza mimic. It mimics all the functions of mycorrhiza. So when I say it mimics mycorrhiza, it has been proved through molecular biology, and it is culturable. Mycorrhiza are not culturable outside the root system, but P indica is culturable outside the root system. So that makes it very unique. We can mass cultivate it. As I speak, industry is producing it in a 1400-liter bioreactor right just now in India. And this technology has been transferred to 6 companies, including one in Taiwan. So P indica, just like mycorrhiza, colonize the roots of the plant, sends its fine hyphae outwards, and the response is not just in the root zone, but in the distal plant shoots also. It enhances flowering, enhances grain yield, provides tolerance under biotic and abiotic stress. As I said, it is isolated from the Thar desert of Rajasthan, but it is equally working in the cold desert of Leh Ladakh, enhancing the yield of the cold vegetables growing in that area. So this technology is very well grounded, very well recognized, very well applicable. Over 1 million farmers are benefiting as I speak. Our next technology, also based on a novel root endophyte, that is recently discovered by our group. I led that expedition from the Kandi region of Punjab, that is Teluromyces albopicillus HNB9, a unique culturable root endophyte just like indica, and it is a very good solubilizer of phosphate, zinc, silica, produces ammonia and oxin, and provides tolerance against biotic and abiotic stress. Particularly, we have validated the tolerance against Fusarium oxysporum, which is a major pathogen, including the race 1 and 4, the pathogen of banana. So we have validated it across, and we are in talks with industry. The basic microorganism, that is HNB9, has already been transferred to a Delhi-based startup, and along with that, we have made a consortium with the incompatible entomopathogenic fungi, Metarhizium and Beauveria bassiana, and we are the first in the world to make the consortium of this Metarhizium and Beauveria bassiana along with HNB9 fungus, and we have transferred this technology also to the company. And this HNB9, when I say it's a phosphate solubilizer, the rate of phosphate solubilization is that it is solubilizing rather more than 40 gram of tricalcium phosphate per liter in a fermenter. So we have patented this technology also. Not only it is solubilizing the phosphate, tricalcium phosphate, it is also solubilizing magnesium trisilicate, zinc oxide also in a medium, and we have made multiple consortium of this fungi with Enterobacter cloacae, that is a very strong potassium mobilizing bacteria. So in effect, we have made a bio NPK, and just yesterday also we filed a patent, sir, on its combination with Pseudomonas
Pseudomonas fluorophis that produces vinagreine compound. Surprisingly, that compound is antifungal in nature, but we could obtain the compatibility and these two synergize together to suppress Fusarium oxysporum and also enhance plant productivity. So yesterday only we could file the patent.
Thank you. I think during lunch you join them and explain more because you are very relevant to their activity microbiota. Thank you. Dr. Sushmita Shukla, she is from Amity Institute of Biotechnology and she works on tissue culture and other related areas for micropropagation and also true to replication is, that is the mother to the offspring. How do you do that? True to replication.
Right, sir.
Over to you.
Thank you so much. A very good afternoon to all the dignitaries of the dais and of the dais. As Salomati sir already spoke about me that I'm working at Amity Institute of Biotechnology. I do have the well-developed tissue culture lab where we have already propagated under robust protocol for about more than 10 species we are having. I have worked, our group has worked on Stereospermum personatum as well. Stereospermum personatum and Stereospermum savolens are the two species where are being used in Ayurveda formulations. The roots are being actually extracted. So there again we are getting the importance why the tissue culture or micropropagation of such three species are needed. So whenever it is required, we can multiply them and we can get the roots in vitro and we can again go for the formulation. Other than that, I have also worked in the endangered citrus species where we got a very good project under DBT twinning project where we have grown the full robust protocol for the different species of citrus and our was the citrus macroptera. So that is a very unique species which is being used for different culinary dishes and all the different and it is being only the rind of that particular citrus are being used, not the juice actually, but it is for making many different pickles and different things. So that also we have already propagated methods gate number 2 in organic farming. It is growing there. We will get time if Prutan ma'am permits, then we can go and we can select that also. Other than that, also we have transformed the Edred gene, where again drought tolerant Vigna species we have transformed very successfully, and it is also there transgenic Vigna radica. There again, it is a leguminous crop which is there. And I was also means the full on protocol what we are doing in a lab, right from initiation till the hardening, we go for and we do produce those particular plants.
Thank you. Thank you, Sushmita. We'll
Thank you
talk
so
to you.
Yeah, I'll like to
Right.
talk to Dr.
Very good. Now I have Dr. Kusma Panda from AIDA. You can mention about your research in this area. Dr. Kusma.
Good afternoon, all the guests present on the dais here and off the dais. So, as said, I'm from Amity Institute of Biotechnology, and my work is on breeding and genetic analysis. Especially, I have done the genetic analysis using the breeding method in millets, that is bajra and sorghum. Especially, I have emphasized on the traits related to salinity tolerance using this F2 analysis. And also, we have worked, it's a collaborative project. We have worked on the chromosomal analysis of all the Indian species and their karyotypic analysis. And I have also worked on starch, resistant starch in different rice varieties, and also characterization of black rice germplasm, especially for its various nutraceuticals and also health-related compounds. And presently, my scholar is working on the salinity tolerance and effects of these biofertilizers and PGPR, especially this rhizospheric and endophytic microbes. So how they will be working towards the salinity tolerance or any other abiotics tolerance in jowar. He's right now he is concentrating on the jowar. That can be also extended to other millet crops. So this is how we have been working. So I was interested, as you have said that AM fungi are involved in the drought and the salinity tolerance. So maybe I would like to coordinate and connect with you.
Very good. Let's clap for Dr. Kusma. Very good. Now I move on to Dr. B.K. Chowdhury. He is also one of our brilliant scientists and who is also a top 2% scientists across the globe as the Stanford survey. And over to you, Dr.
Thank you, sir.
Chowdhury.
So nice of you. I will not take much time. 3 or 4 projects all together we operated earlier, sponsored by DBT, DST, and SERP, and wherein we provided emphasis to see the benign impact of the Pseudomonas bacillus strains, to see the induced systemic resistance, induced systemic tolerance against microformula of Pasolina, also called in toto biotic and abiotic stresses. So we filed 3 patents also, and we have already been granted these patents, and we are in collaborations with some technology transfer also. That is my gist, sir. Thank you.
Thank you, Dr. B.K. And I will move to Dr. Neetu Singh, who is our extension person, who has done a lot of training of farmers. And over to you, Dr. Neetu. Just highlight a few of the important extension activities. Agricultural extension.
Okay, thank you, sir. And good afternoon to all dignitaries on the dais, especially our guests. Welcome to Amity. Myself, Dr. Nitu Singh, and our DG already explained about our center's name. In our center, Amity Center for Biocontrol and Plant Disease Management, we are working on the theme like field to lab and vice versa studies. So we have already worked for a lot of soil-borne diseases, particularly major like root knot nematode, wilt, and rot fungi, and disease complexes also. Or in for the management of these diseases, we have developed several packages. These packages are known as the IPM packages, Integrated Pest Management Packages, and comprising of a very sustainable components like oilseed cake, botanicals, and biofertilizer and biopesticides also. And these packages are very popularized among the farmers. They adjust to it, particularly for horticulture crops. And these IPM packages popularized in Western UP also, in Haryana also, and farmers are getting benefited because these packages are as an alternative to chemical farming. Because a lot of chemicals farmers are using out of ignorance, so we have popularized. And apart from these packages, we are also popularizing oilseed and pulses crops under National Food Security Mission in Western UP, particularly Gautam Budh Nagar and Bulandshahr district. So farmers are also benefited with the different type of crops also.
Good. Let's clap for you too. Very nice. Good work that you are doing with farmers. And Arpita, she is also from biotechnology. Can you just pass the mic to her? Then I would request Dr. Kotari, if he is there online. And can you switch on your camera, Dr. Kotari? From
Yes, sir. There is some problem in my camera, looks like. I have switched it on. Can
Sir,
you hear me, sir?
Sir, your laptop shutter is off, so you have to just.
Can you hear me, sir?
Yes, sir, we can hear you.
The camera, there is some problem.
Sir, you
So,
have to open
okay.
the shutter of the camera.
My camera is automatically switched on, but picture is not coming.
No problem. Next. Okay. Now, Arpita, can you finish your comment? Then I'll answer.
Yeah.
But can I have some interaction without the camera?
Yeah, please. Hold on. Just 1 minute you give to Arpita, then I will request you, Kotari.
Okay, sir. Right.
Thank you, sir. Good afternoon, all present here. Myself, Professor Arpita Bhattacharya. My specialization is in nanotechnology and chemical technology. Currently, I am working on nano packaging, the post-harvest management, and the nano coating with the essential oil specifically. And my area is encapsulation. So, with the nanotechnology, whatever the different part of the agriculture and horticulture, the different research I am enriched, like the effect of the nanomaterial, different types of nanomaterials for the pathogenic fungus control, nematode control, and different formulation with these nanomaterials. These are my.
Thank you. Thank you, Gita. Very good. Good work what you are doing. Now, Dr. Kothari, can you make a brief comment on Amity University, Rajasthan, Jaipur?
Yes.
Agriculture related, plant related research, if you have.
Yeah, at Jaipur, we have been working on plant tissue culture and effect of nanomaterials on plant structure and function and plant morphogenesis. In several plants, we have observed that. Can you hear me, sir? Am I audible?
Yes, we can hear you.
Yeah. So in several plants, we have seen the effects of nanomaterials such as carbon nanotubes, silicon oxide nanoparticles, silver and gold nanoparticles. They have some positive effect on the growth and development of plants. Using silicon oxide nanoparticles, we have seen that in some of the leguminous plants, when the seeds are treated with nanomaterials and then the seeds are sown, they have larger number of nodules and their nitrogen fixing ability is also enhanced. Biomass production is also more. In Madam's lecture, what I saw was effect of mycorrhiza on the growth and biomass production in plants. If we use some of these nanomaterials, nanoparticles, carbon CNTs or carbon nanomaterials and other nanomaterials and nanocomposites, some positive effect I think should be there. So I do not know if anybody has used nanomaterials and their effect on the mycorrhiza and associated plant growth and development.
Thank you, Dr. Kothari. Kothari is the Vice President of Amity Science Technology Innovation Foundation. He is in Amity University, Rajasthan, Jaipur. So among the 12 universities, there is distinguished university. He has established a big R&D facility there through PERS program, which he got from the Government of India. So thank you so much for coordinating and giving a new impetus to research.
Thank you, sir.
We have almost 25 to 30 people online listening to all your lectures, all our deliberations.
Sir, I would like to say through you, if both the speakers have time and if they want to visit Jaipur, it will be our pleasure to host them and have their lecture and interaction there at Jaipur Amity campus, which is very near to Delhi.
It takes only 4 hours from here. Is that right? Yeah, maybe doing this later, but Diana was telling next time. Okay. Thank you so much for this invitation. We appreciate your gesture. Now, thank you so much for the distinguished delegation. Now, I like to have their feedback. You listen to all our scientists, faculties, and their zeal and enthusiasm to work with you, with the Hungarian institution. So, how would you look at expanding this partnership? And many of our students have gone to your university, Szeged. And also, we have already begun, Seema Garg has been working with Hungary and also including your institution. So I'd like to hear your views, aspirations, and guidance that how do we move forward from this collaboration. Now, I will start with Dr. Diana first.
Thank you, Dr. Selvamurthy, and thank you for all the warm welcome. And also, I would like to thank all the faculty members to offer their holidays for us and have welcoming us and have a discussion on possible. Okay, closer. Okay, possible. Wrong. Either I am too long or the distance is wrong. So whatever. But I wish to express my sincere and deep thanks for everyone who is present here to discuss the possible collaboration with Hungary, especially with the University of Szeged, from which two researchers are here. And I do hope that we could enhance not in just the field of agriculture and the climate and what already Dr. Seema Garg mentioned, but in other fields, we probably find synergies and possible cooperations. And I do hope that we also find the funding for the mobility, for not just for professors, but also for students. Thank you so much.
May I have the views of Professor Thomas?
So, thank you once more for your hospitality and the possibility to introduce a part of our work. And thank you very much for the introduction of your work, that is very, very diverse. And here I must add that at our university, that has 12 faculties, not our faculty is the only one that is involved in agricultural research, but also the faculty of science, faculty of engineering, and faculty of pharmacy are involved in agriculture-related works. Usually, the setup is that the innovations from the microbiology side come from the Faculty of Science. They work with really the microbes, and they make the composition of preparates, and we are the appliers, so who test how it is working in real life. So that is the usual setup, but Dr. Melinda is conducting mycorrhizal science on her own with colleagues. She will tell some words about it, already talked to. So this is one side. There is the Department of Plant Physiology, Plant Biology at the Faculty of Science are making basic science in plant physiology. We usually focus on the applied part, but now we've got a young colleague who is introducing what is bioimpedance. Bioimpedance to test the various traits. And so now we went also in this direction to test plants from this side also. And Dr. Melinda is involved also in the application of AI in the test of plant stresses. So here the collaboration with the IT guys of our university is very developed. So I think it will be fine to have this as a material in a written form or something in electronic form, because we could not make notes that much about your short talks. But I think there are numbers of fields for collaboration. Nanotechnology is another thing. Nano guys are involved from environmental protection to even plant production, because one of our colleagues at the plant biology department, she is developing a kind of seed coating material to with the application of nanomaterials. So numbers of fields are open besides the already existing or in the future existed collaborations with Professor Clara Hernadi and the other colleagues from our university. And we look forward for this period when we can generate common projects either in the frame of international programs. I always say when I have to discuss about international collaborations that science is always the easiest field. So because we work on something here and we are working on something similar in the same context at home, and then we fuse our results and as the first step it usually works. And then whether the mobility and the visiting is possible, is it supported or not by any means, hopefully yes, then it's another question. Okay, thank you very much.
Thank you. Thank you, Professor Thomas. Let's give a big clap to Dr. Thomas. I think we have already mentioned some of the ways by which we can expand this collaboration. Now, I would like to listen to Dr. Melinda. What are your aspirations for collaboration, Dr. Melinda? Because you also listen to some of your counterparts in Amity University here.
Thank you very much, Dr. Savamurthy. Thank you for the invitation and for the collaboration possibilities with you. I am very happy to hear your peers and your brief introductions. And as a researcher, I'm very open for the collaboration with you and also with the students, PhD students. So I hope in the future we will have a fruitful possibilities for collaborate together and write projects and papers and so on. So I'm very happy to meet with you. Thank you very much.
So with that note, let me conclude just summarizing what we can do together. The first thing is the just received a message from to just read out to our delegates. See, now he has announced a mission, Global Synergy Connect. There is a mission connect, but let's give a big clap to founder president. We are coming here, there is a new mission launched, Mission Global Synergy Connect. And this will not only research, academic, student exchange, faculty exchange, and internationalization in a big way. So this will be a very important initiative. Now, our colleague from AUUP is here, but we should now take it forward. Now, I would suggest the first is we will have a joint record of discussion, what has been deliberated in this visit. And we will send it in a draft form, Shefali, and to them, to Ambassador, to our Science and Technology Counselor, and we will send it to the embassy and then get it issued, jointly signed as a joint record of discussion. From here, Nutan Kaushik can sign, and so from your side, Ambassador, the Science and Technology Counselor, so we can sign together so that there is a record of discussion which you can show to your government and we can show to other. That's the first step. The second step is I would request within a month. No, the second step is we will send all the presentations which were made, including CMA, to our Dr. Diana, so that she will know the span of activities, including my presentation, so that she will be able to connect when such distinguished faculties visit from various universities in Hungary. Then she will be able to connect. There is an institution, so many scientists are working. So that second part will be send all the presentation to her after duly seen. Then the third will be arranged within 1 month, that after they have ruminated, what we are going to do, step 1, 2, 3, 4, milestones. Milestones, what we can do together this year, 2026-27, make a roadmap together. That will be formulated in a Zoom meeting, which Nutan Kaushik can coordinate within a month, so that there is a follow-up which will take place. So this you can select who are the representatives who would be required to be present, and from your side you can nominate maybe even more than two of the professors here. So this will augment, and then maybe after 3 months again we will review, so that there is a pressure on the system to perform outcome, which founder president keeps talking about. So this would be the process. So we would like to see when by end of 2026, some of the program would have taken off together, more programs. So we would like to know by end of December 2026, when we review, so many students have gone, so many faculties are going, and so many projects have been formulated and submitted in the bilateral, multilateral schemes available. So this is how if we do, there will be a fruitful outcome. So once again, I thank you. Let's give a big clap to Dr. Shefali Kashyap. Shefali Kashyap, before we ask a formal thanks, I want to thank the FCR team, particularly Dr. Shefali and also Dr. Shweta Agarwal and Kavita Joshi and also the IAD team. So yeah, and Meghna, Dr. Meghna is here from team and everybody. Yeah. So Shweta will coordinate with it. So yes, Shweta. So let's give a big clap to Shweta. We have big responsibility now to do. We will ask you what all you have achieved. So make it happen. I'm sure we'll make it happen. And I must compliment all my faculties, scientists who are here. You are doing a great job for not only the country, for the humanity. So realize when the demand on food is going up with growing population, and also the cultivable land and the soil, climate change, there are so many challenges for us to address. So I'm sure all of us in Amity University, all those who are present online, we will resolve that, we will work together with Hungary and other countries, because India has always been the global village, global family, and also Vasudeva Kudumbakam, that we are all one family, brothers and sisters. We speak different language, maybe having some boundaries, and we have some colors, different colors, but we are all one. So that is what India has always cherished, Indian philosophy. So with this, we will resolve to do work together as one family, one scientific family, to address all the needs of the humanity, including agriculture and food. So with this, I will now hand it over to. So let's give a big clap to Shefali, who is coordinating the whole event. Now over to you, Shweta, to give a formal word of thanks.
Thank you, sir. Good afternoon, everyone present. On behalf of Amity Foundation for Science, Technology and Innovation Alliances and the S&T War Room, I would like to firstly extend my sincere gratitude to our Honorable Founder President, sir, for all his guidance and blessings. The success of this event would not have been possible without his unwavering support. It is the inspiring vision of our Honorable Sir that Amity expands its global footprints and further strengthen the bilateral collaboration in the domain of research, innovation, technology, and science. My sincere thanks to our esteemed guests, Professor Thomas and Dr. Melinda, for sparing their valuable time and for sharing their insightful deliberations and expertise in sweet potato research and effects of biofertilizers on plant production and soil health. My special thanks to Dr. Diana from Embassy of Hungary and Professor Dr. Gurinder Singh for their continuous support and encouragement in strengthening Indo-Hungarian partnership. My heartfelt thanks to Dr. Silvamurthy sir, Group Captain Kapil Shukla sir, Dr. Nutan Kaushik ma'am, Dr. Doily Sanyal ma'am, Dr. Meghna, Dr. Shefali Kashyap, Ms. Kavita Joshi for their motivation and support. I would also like to acknowledge the valuable contributions of all the faculty members, researchers, scientists, IT and admin team, ESCO and media team, and my colleagues, whose effort made today's program enriching and successful. Now we look forward to building stronger collaborations with the University of Szeged and Hungary as a whole in times ahead. Thank you once again to everyone for your gracious presence and participation. Thank you.
Good. Let us clap for Shweta. Shweta, you did a wonderful word of thanks. You covered everything. Good.
May I request now Ms. Kavita Joshi to organize the felicitation of the guests.
You are working well. Is this on?
Thank you.